Welcome to the weekly Neurology Deep Dive, produced by Amer Gavanini. We take the latest, most impactful research and break it down for you. We're always focused on the essential, actionable information you need for your clinical practice.
And before we begin, just a quick note. This deep dive is a summary for educational purposes only. We have no commercial goal here. Given the detail we're about to cover, we really do recommend that you read the full paper for the complete context.
Today, our mission is to cut through some of the noise surrounding Myelin Protein Zero, or MPZ-related neuropathies. Specifically, we're looking at Charcot-Marie-Tooth type 1B and a fascinating paper that correlates the molecular mechanism of a variant with the patient's actual clinical severity.
Let's get right to the most practical takeaway. For you, the busy neurologist, the key word today is stratification. We have to stop looking at MPZ-related Charcot-Marie-Tooth as a single entity.
Because this paper shows pretty definitively that these MPZ variants can be split into two categories. You have the destabilizing variants and the non-destabilizing variants.
And the clinical difference between these two groups is, well, it's monumental. The destabilizing variants, the ones that trigger a kind of cellular crisis, they lead to disease onset almost 5 years earlier and are linked to substantially worse neurological impairment.
So this is now mandatory information for prognosis, and more importantly, for therapy. If your patient has a destabilizing variant, they might be a candidate for some of these emerging, very specific treatments.
Exactly. Whereas if they have a non-destabilizing variant, you need a completely different therapeutic mindset.
Okay, let's unpack this a bit. We should probably start with the molecular context.
Right. We all know Charcot-Marie-Tooth is the most prevalent hereditary neurologic disorder. In MPZ variants, they only account for a small slice, somewhere between, say, about 2 and 10% of cases, but they cause this incredibly wide spectrum of disease. And MPZ itself is just foundational. It makes up about half of all the protein in peripheral nerve myelin. Its whole job is to, dine to itself to zip up and compact the myelin sheath. When it goes wrong, you get demyelinating neuropathy, which is usually Charcot-Marie-Tooth type 1B.
But the key insight from this paper is that how it goes wrong seems to matter more than just that it went wrong. Why don't you walk us through those two different failure modes they found? Let's start with the more severe group.
That would be the destabilizing variants. These mutations are deep inside the protein core. When a mutation happens there, the whole protein misfolds. It gets mangled. And this malformed protein starts accumulating in the endoplasmic reticulum of the Schwann cell.
So it creates a sort of cellular traffic jam overwhelming the quality control system. And that buildup triggers the cell's alarm bell, the unfolded protein response, or UPR.
Precisely. The UPR is this universal stress signal. When it's firing constantly, you get toxicity, inflammation, just widespread cellular dysfunction. This is what we call a gain-of-function mechanism. The mutated protein is actively poisoning the well.
Okay, so if that first group is actively toxic, how does the second mechanism, the non-destabilizing variants, cause disease?
It's a totally different story. The non-destabilizing variants are on the surface of the protein, in the extracellular domain. That's the part that's supposed to reach out and grab onto its neighbor. These mutations don't cause misfolding, and they don't trigger that UPR stress response.
So it's less of a toxicity problem and more of a structural engineering failure.
That's a perfect way to put it. They just impair MPZ's ability to bind to itself, what we call oligomerization. So the proteins can't stick together properly, and the myelin just can't be compacted correctly. It's a dominant negative or loss of function mechanism. The cell isn't panicking. The structure is just poorly built.
And that distinction is so crucial because it immediately points to two completely different therapeutic roads. We've already seen some powerful preclinical data on targeting that UPR pathway.
That's the whole therapeutic motivation here. You think of agents like Cephin-1, it's a small molecule that modulates the UPR. And in animal models of Charcot-Marie-Tooth type 1B, it has shown real benefits improving myelination and motor function.
But the clinical translation is everything. If a patient's disease is from that structural failure, the loss of function, then a drug designed to calm down cell stress, like Cephin-1, is probably not going to do much.
That's the entire point.
Yeah.
We need to know who's having the cell crisis and who's just suffering from a structural deficit.
Which brings us to the study itself. To figure out who could benefit from these UPR modulating therapies, they needed definitive clinical data. How did they set this study up?
It was a retrospective cross-sectional analysis. They pooled data from 7 different Italian referral centers, which is a pretty significant effort. They looked at 75 patients, all with a confirmed MPZ variant. And the key inclusion criterion was early onset, so the disease had to manifest before 18 years of age.
Now, classifying every single variant based on lab data alone would be a huge challenge. How did they actually sort them into the destabilizing and non-destabilizing buckets?
They started with published functional data where they could, but for many of the missense variants, they had to turn to computational analysis. They used an in silico tool called DOAT.
And DOT calculates this value, the change in Gibbs free energy, or delta G. For the clinician, what's the value of that computational approach?
It lets us predict pretty quickly the structural fallout of a mutation. A more negative delta G value suggests a higher degree of protein destabilization. So the computer model basically predicts how likely the protein is to collapse on itself and trigger that misfolding cascade we talked about.
Looking at the 75-patient cohort overall, they were certainly a highly affected group. The mean age at onset was just about 4.7 years.
And you can see the disease burden. The mean Charcot-Marie-Tooth examination score, or CMTES, was 9.5. Importantly, they confirmed this is a progressive disease. There was a significant positive correlation between the CMTES and the patient's age at assessment.
The average progression rate for the whole group was about 0.11 CMTES points per year, so not a static process at all. They also noted the most common variant was PS78L, which was found in about 43% of these patients.
Right. Now let's get to the core findings. The head-to-head comparison between the destabilizing and non-destabilizing groups. The differences start with onset and inheritance.
And the age difference is just staggering. The destabilizing group had a mean age of onset of only 1.4 years. Compare that to the non-destabilizing group, where onset was significantly later, averaging 6.7 years.
That's more than a 5-year difference. It completely changes the pediatric management conversation.
Yeah.
And to really underscore that severity, infantile onset, so between 0 and 5 years, was seen in about 95% of the destabilizing group.
We also see a really interesting difference in how these variants arise. The destabilizing ones were about 57% sporadic, meaning they were mostly fresh de novo mutations.
Which suggests the disease burden is so severe, it might actually impair reproductive fitness over generations. By contrast, the non-destabilizing variants were about 94% autosomal dominant, so they're more successfully inherited. That difference was highly significant.
And when you look at the clinical severity measures, the actual functional impact, the gap between the two groups is unmistakable. All the indicators were significantly worse in the destabilizing group.
Let's translate that into what it means for the patient. For ambulation, in the destabilizing group, about 20% of patients needed a wheelchair. In the non-destabilizing group, 0.
And conversely, independent walking was only achieved by about 29% of the destabilizing group versus about 76% in a non-destabilizing group.
The need for supportive devices tells a similar story. About 47% of the destabilizing patients required ankle-foot orthoses, or AFOs.
Yeah.
That's compared to only about 15% of the non-destabilizing patients.
And this severe early onset group also had more global involvement, right? Proximal weakness was more common.
Yes, significantly so. Proximal lower limb weakness affected about 41% of the destabilizing patients, compared to only about 8% in the milder group.
We can't forget the upper limbs. Difficulties with hand motor skills were almost universal in the destabilizing group, about 95% of patients, versus about 45% in the non-destabilizing group.
Even secondary complications like scoliosis were significantly more frequent, found in about 63% of the destabilizing group, compared to about 18% of the others. And sensory loss was profound. Paresthesia, or vibration sense, was severely impaired in about 62% of the destabilizing group versus about 22%.
And if we bring that all back to the Charcot-Marie-Tooth examination score, the average severity score was 12.1 in the destabilizing group versus 8.2. But I think the most telling figure of all might be the rate of decline.
The data really suggests an accelerated course for that UPR activating group. The calculated progression rate was 0.24 CMTES points per year for the destabilizing group. That is more than double the 0.11 points per year seen in the non-destabilizing group.
So for the clinician, this suggests that this group of patients is facing functional decline at twice the speed. This high rate of decline makes the molecular insights even more urgent. Let's go back to that computational analysis with the delta G values. Did the prediction hold up against the clinical data?
It did, and very strongly. After they excluded one particular biological outlier, the delta G value showed a strong negative correlation with the CMTES. In other words, the greater the predicted protein collapse, the greater the clinical severity. and that correlation was highly significant.
Okay, we have to talk about that one crucial exception, the PY82S variant.
Yes, this is a perfect lesson for translational neurology. The computer predicted that the PY82S variant should be highly destabilizing. It should have been toxic. However, previous in vitro biological work had already shown that this specific variant does not activate the unfolded protein response pathway.
So, at the end of the day, the cell's biology trumps the computer algorithm. These computational tools are powerful for screening, but they have to be validated by the underlying cellular process.
Precisely. It means you really have to integrate everything, the genetics, the computational prediction, and the functional biology, to truly classify the patient.
So what does this all mean for the neurologist in the clinic today? How do we use this mechanism-based classification?
Well, it directly dictates your therapeutic strategy. For patients with those destabilizing UPR-activating variants, therapies targeting that stress response like 7-1 are highly relevant. We should be prioritizing these individuals for potential UPR-based treatments and trials.
But for the other group, the non-destabilizing, loss-of-function patients, we need a completely different playbook.
For them, you have to think about restoring function or removing the bad protein. Things like variant-specific allele editing, gene silencing, or maybe even replacement with a healthy allele. The treatment has to be custom-fitted to the mechanism.
And finally, this gives us a critical parameter for running clinical trials. That accelerated progression rate in the destabilizing group, 0.24 points per year, that could be a huge advantage.
It's vital for rare diseases. It means you could potentially show a treatment effect over a shorter period of time with fewer patients. Going forward, trials for UPR-modulating drugs should really focus on children, even pre-symptomatic individuals, who are known to have these destabilizing variants.
Let's just quickly recap that primary clinical take-home message one last time.
MPZ variants can be reliably stratified based on their molecular effect. Destabilizing variants activate the unfolded protein response, leading to a severe, earlier onset disease with faster progression. Non-destabilizing variants cause a loss of function due to structural failure, and that presents as a milder, later onset disease. This stratification is now essential for prognosis and for selecting the right therapy.
I think this deep dive really shows that classifying these inherited neuropathies based on the actual mechanistic effect of the variant, is it a protein folding failure or a protein adhesion failure, provides much stronger clinical segregation than just relying on broad categories like age at onset.
Absolutely. The future of treating these diseases lies in that focus on mechanism. We're moving beyond just diagnosing the syndrome and toward diagnosing the specific molecular pathway failure that needs to be corrected.
Thank you for joining the weekly Neurology Deep Dive. We look forward to exploring the next frontier of research with you soon.